We're going to start with the yolk sac because that is the earliest stage of these three phases that we're going through. And this occurs very shortly after implantation. And during this journey down the uterine tube, these cells that were dividing created a hollow vault. So the interior of this group of cells is hollow. And that hollow chamber is called the blastocyst, and on the 8th day after implantation, it begins to take on an appearance like this, and then it develops into an amnion, which is what this illustration is showing you about the second week, and eventually forms an amniotic sac. Now the amniotic sac is a membrane that surrounds the developing group of cells, this embryo, and is going to become kind of an insulative chamber for the fetus, embryo and fetus to develop. Amniotic fluid comes primarily from the plasma components of the mother's blood, and later fetal urine is also included. The sac functions as a shock absorber, fetal temperature regulator, protects the fetus from abrasion and dehydration. So it's like a little cocoon, if you will. It's the amniotic sac that ruptures when someone says your water has broken. It's the amniotic fluid that then leaks out, and that generally leads to contractions. The placenta is the junction between the mother's circulation and fetal circulation. So if you look at the uterine wall here, as the fetus develops, two separate groups of tissues form. There's some fetal tissue that's half of the amniotic sac, which leads from the umbilical cord to the fetus. And then there is a second layer of tissue inside the uterine wall, which the mother's circulatory system is hooked up to. And this then is the transfer point for nutrients and waste products. So the circulation of the fetus goes like this, and the circulation of the mother goes like this across this intermediate membrane. And across this membrane can travel glucose and oxygen and carbon dioxide and urea and other urine-type waste products. Antibodies can pass across this membrane, but cells don't generally pass across a membrane. So we're not combining the circulation of the fetus with the circulation of the mother directly. It's an indirect relationship across this membrane of the placenta. And it's the development of the placenta then that's different, that makes us able to bring a developing fetus to full term. Prior to that, if you're to look at marsupials, for example, like possums, they don't have a placenta, and therefore their young are born at the end of what would be the first trimester in a pouch, and they then develop in that pouch over a period of time. But we are the placental mammals, so that enables us to bring a fetus to full term. And it's this relationship between the mother's circulation and the baby's circulation that enables that. So the placenta is a pancake-shaped structure, one side of which is produced by the mother, the other side by the fetus. It is attached to the uterine wall, and the mother's side to the umbilical cord of the fetus side. The placenta becomes fully functional at the end of the first trimester. And then, of course, during childbirth, once the child is born, the placenta, both sides, both parts of the placenta, peel off the uterine wall and come out with the umbilical cord. Every once in a while, you may have a problem with the placenta beginning to tear off of the uterine wall prematurely. And if that happens, that's a very critical situation because that cuts off the oxygen supply to the fetus, and something has to be done very, very quickly. But generally speaking, that's not an issue. So here's an illustration of a little bit later. This is at 13 weeks. You can see the amniotic sac, the amniotic fluid, the umbilical cord, and the placenta, all represented there. One other thing, or a couple other things actually. If you have some concerns genetically about the developing fetus, there are a couple of samples that can be taken in fetal development to determine the genetics of this particular fetus. One is called amniocentesis. It's been around quite a while. And what you're doing is actually extracting some of the amniotic fluid. In the amniotic fluid are some fetal cells that you can get genetics from, or genes from, or chromosomes from. A little bit later, I think that's been around, what, 25 years or something like that. And then the chorionic villi sampling is a little more recent. It can be done a little earlier. It actually samples the chorion, or the tissue around the fetus, which also has some fetal cells, which can be used to determine the genetics of the offspring. So those are two genetics tests.
Excuse me, Mr. Tone, so that the chorionic villi sampling is taking cells from the baby side of the placenta?
Yes. Now, there are a lot of embryology is really fascinating stuff, and there's a lot of things that go on that are really interesting. But one particular thing that I think is important to understand is fetal circulation is quite a bit different until the end of the third trimester than it normally would be, and that is for a couple of reasons. While the fetus is developing, there really isn't a great need to circulate blood to the lungs, other than enough blood to have the lungs develop, and enough blood to the kidneys for them to develop. Because during this time, it's the fetal circulation that's supplying the carbon dioxide to the mothers across the placenta wall. So the lungs aren't getting rid of the CO2, the mother is. The mother's receiving urinary waste products, so the kidneys aren't typically functional. And because of that, there is a space that is open between the two atria of a fetal heart. And if you remember the circulation of the heart, blood comes in the right side and then gets pumped to the lungs, then comes over to the left side and gets pumped to body tissues. Well, what happens here is blood comes in the right side, it goes right over to the left side into body tissues, short-circuiting the pulmonary circuit, because pulmonary circuit's really not functional. So it kind of just short-circuits that. But toward the end of the third trimester, this opening, the foramen ovale, grows back, or doesn't grow back, grows together to seal off the two atria, so when the baby is born and begins to breathe, then there's going to be adequate blood supply going to the lungs so that they can exchange gases. And similar development takes place with the kidneys and some of the other organs like the liver as well, once they develop to the point where they can become functional. But that's really toward the end of the third trimester that occurs. And like I said, there's a lot of other things as well, but that's one of the more. And occasionally, particularly in a premature birth, you may have a problem with that because that tissue may not have developed fully, and that leads to a lot of issues once the baby's born. We're moving on now to sexually transmitted disease and birth control, because there's some relationship between the two in some cases. Sexually transmitted diseases I've broken down into the categories based on the type of organism that causes them, or vector that causes them. The first group are bacterial STDs, and as I hope you know, that antibiotics work very well on bacteria, but they don't work on viruses. So there are developing antiviral drugs at this point in time, but basically it's your immune system that deals with viruses to a greater degree than some sort of prescription. But bacterial STDs can be treated with antibiotics, and therefore you can generally get rid of them. The most common one is chlamydia, and I think that number is outdated. It's more like 6 or 7 million a year are being infected with chlamydia. In females, it can lead to pelvic inflammatory disease. One of the unfortunate things about chlamydia is that it can be asymptomatic for a period of time. In other words, you're not aware of the fact that you have any symptoms, but you're still capable of transmitting it. This is particularly true in males. So a male may be unaware of the fact that he has contracted the disease, but is passing it from partner to partner even without his knowledge. Gonorrhea and syphilis are a little bit less common. Every once in a while you get some antibiotic-resistant strains that show up that become problematic. There was one on the West Coast in 2004 with syphilis. But to a great degree, these are quite treatable once they're diagnosed. Viral STDs, generally, if you contract them, you have them for the rest of your life. Whether they're active or not may be, maybe a situation, but you will possess them probably for the rest of your life. AIDS, of course, is the most devastating one. I understand in October there's going to be an AIDS test available on the market for about $50 or something like that. So that might be of great benefit. It invades the helper T cells, as we talked about when we talked about the immune system. Herpes has dormant and active phases, and during dormant stages it's very difficult to physically detect. HPV, that's why we have pap smears in females, human papillomavirus, and it can in some cases lead to cervical cancer, so it needs to be very closely monitored. Parasites are generally not an issue, except in third world countries, because they're quite easily treated. Now, birth control. There are some types of birth control that not only prevent conception, but also help prevent STDs from being transmitted from one party to the next. And probably the most significant one of those are the latex barriers like condoms and diaphragms and cervical caps and vaginal pouches. Those are all relatively effective birth control, but they're also reasonably effective. They're quite effective against bacterial STDs, but they're not 100% effective against viral STDs, because viruses can pass through a latex barrier. It's going to restrict the chances or increase the chances that that's not going to happen by quite a bit, so it's well worth the effort. But don't be lulled into the idea that a latex barrier is going to prevent a virus from passing, because viruses are small enough to pass through a latex barrier. The most common of all of these is the male condom. And most of these are quite effective. Maybe not as effective as you would prefer, but they're about 90% effective when properly used. However, sometimes they're not properly used, so the average effectiveness is about 80%. There is another birth control method that was attempted to a significant degree in the 60s and had so many side effects that it was kind of dropped, but now it's reappearing, and that's the IUD. And the IUD is a actual thing, like a plastic device that you actually insert in through the cervix into the uterus, and it physically prevents implantation.
Sometimes.
Well, yeah.
I'm sorry. My niece actually heard us that out, and she's expecting now, but yeah.
As long as it remains in there,
Yeah. It's not supposed to come out.
it's not supposed to come out. That's right. Well, there's always kinds of strange things that occur.
Oh, yeah.
But the IUDs of today are not nearly have the side effects that they used to have. And the next group that were sponges and foam used in concert with condoms and latex barriers, increase the effectiveness of them because they basically immortalize the sperm so they cannot fertilize an egg, so that increases the probability of effectiveness. But as you probably well know, that the choice of birth control for most females is the pill, is a hormone-related substance like the pill or Depo-Provera or progesterone implants, all of which increase the estrogen and progesterone levels in the blood, which stop ovulation. What most people don't realize, and I don't know if gynecologists are not good at communicating this or not, is the pill is not a long-term solution to birth control, because over time the pill in many people has lots of side effects. So it should be considered a temporary thing for less than 10 years, probably less than 6 or 7 years, and so that's where the IUD might play a bigger and bigger role as long as they remain in place. The pill is a daily dose with a placebo at the end. The injections, Depo-Provera, can last about 3 months. Progesterone implants can last up to 5 years. The longer term dosages, the downside is if you decide you want to get pregnant, you're going to have to wait quite a while for them to leave your system. So it's going to be a while before you're fertile again. The morning after pill should be taken within 72 hours of intercourse, and it operates differently based on the timing. If you take the morning after pill immediately after intercourse or within 24 hours of intercourse, it's most likely going to prevent fertilization. If you take it after 24 hours, it'll prevent implantation. So it's a question of timing because of what's going on inside your body. RU46 is used in Europe but not approved in this country, but it can be used up to 7 weeks after intercourse. Then there are physical methods like rhythm, which is periodic abstention during ovulation. If you have a very, very consistent 28-day cycle and you use the simple thermal method, which is checking vaginal temperatures and secretions, you can get up to about an 80% effective rate, but that's not a very desirable effective rate. And then there's also the old withdrawal method, which depends on males, and you know how dependable they are. So, of course, you can choose abstention. That always works, okay? No fun, but it always works.
Lord of Jesus.
Surgery should be something that you decide to do if you positively are sure that you don't want to have children ever again. And you have to think about what life-changing things might occur in your future before you make that decision. These percentages they're about 99% plus effective. The successful reverse rate has gone up significantly. I need to change this because vasectomies are probably about 85% reversible now, not 50. And tubal ligations are about 50% reversible now, not 10%. So there is the possibility of reversal. In a vasectomy, it can be done as an outpatient because here's the scrotum, here's the testicle, here's the vas deferens. They can just go in through the scrotum, snip the vas deferens so that sperm can't find their way inside the urethra and be ejaculated. So that prevents sperm from being ejaculated. A male still ejaculates normally because most of the fluid that's ejaculated comes from the seminal vesicles and the prostate. So you're only losing about 20% of the total volume. So it doesn't affect anything other than fertility. The sperm are simply reabsorbed by the testicles if they're not ejaculated, which they won't be. A tubal ligation requires outpatient surgery generally because it's inside the body cavity. They used to perform these right after childbirth because, you know, the patient's always there and all that stuff made it easier, but they've come to find out that because there's so much change in tissues after a child is born that sometimes those tubal ligations grew back together. So they typically don't do that right after childbirth. They'll do it a few months after childbirth. So you can prevent the probability of reversal of them growing back together. But they are very effective. They then end your need for hormones and all other things that can cause side effects. Any questions about any of that there stuff? I have another video for you. I can't remember if I showed it to he is born. His cerebral cortex is not connected to the rest of his central nervous system. And the cerebral cortex, the cerebrum, is responsible for thought, responsible for coordination, responsible for all those things that you normally take for granted. And what this video does is it gives you a picture of what a child is like when it's first born. Its hearing is way off, its vision is way off. It doesn't have the ability to perform tasks because it's not hooked up to its cerebral cortex. And over the first 12 months, millions and millions of connections are made, making that connection between the brainstem and the cerebral cortex. And so by the end of the first year, this infant is now in charge of itself and can control itself.
So in human beings, is that just in human beings or is that in nature too? Because like a lot of other animals, as soon as they're born, they can walk, run, they have to, know how to function in the environment. So is that just humans that the cerebral cortex isn't connected?
The higher you go up the evolutionary scale, the more disconnected it becomes. And the further you go down, the more connected it becomes. Because the cerebral cortex plays a lesser and lesser role in lower forms of life. So there's no specific answer there, but we are much more disconnected than most other animals. So anyway, this is from the Body Stories series.